As we begin our look at the muscular system, we're going to start by going in and looking in detail, microscopic detail, in the inside of a muscle in cross section. So if you cut through a muscle, an entire muscle organ here, you'll see that there are layers of connective tissue I've indicated by different colors. And the blue on the outside is what we call an epimysium. Epimysium, an outer connective tissue, dense connective tissue covering on the muscle. Inside of that, the muscle tissue is divided into groups by another layer of connective tissue. These areas in here are called fascicles. And surrounding each of the fascicle here in brown, that layer there, and in between here, surrounding each of the fascicles, is what we call a perimysium. And then within each of the fascicles, we have a number of muscle fibers indicated in red. Remember, our skeletal muscle units are muscle fibers, not muscle cells. So we have a lot of muscle fibers in here. And in between those muscle fibers, within a fascicle, I've indicated in green here, a connective tissue. This is a loose connective tissue. It's not the dense connective tissue that forms in the epimysium or perimysium, but a loose connective tissue between the individual fibers, and we call this an endomysium. So that's a cross-section of a muscle belly, the belly of a muscle in cross-section. It's got an outer layer, it's got an inside divided into fascicles, and around the fascicles we have connective tissues forming a perimysium, and then within the fascicles we have the individual muscle fibers, and between the fibers we have a little bit of loose connective tissue, that endomysium connective tissue. In this picture on the board now, I have what represents a cross-section of a single muscle fiber. So that would be the outer membrane around the muscle fiber, a couple of the nuclei in the muscle fiber, and then the rest of these little dots I have in here are to represent mitochondria that are found kind of sandwiched between myofibrils. And this sarcomere units are arranged end to end in long strands called myofibrils that would go through here. The next thing we're going to put in here, we'll put in some transverse tubules. Let's go ahead and do that now. The transverse tubules extend from the outer membrane and down in between the sarcomere units and myofibrils down into them toward the center of the bundle of muscle, or I should say toward the middle of the bundle of myofibrils. So transverse tubules will stake their way in here, and there would be a lot of them, more than what I'm drawing here, but just to give you a taste of what the transverse tubules would be like. Also in here would be a number of what's called sarcoplasmic reticulum chambers that surround really each of the sarcomere units. So here in green, kind of outlining each of the sarcomeres with this sarcoplasmic reticulum. We won't do all of them, but we'll put a bunch of them in here. Sarcoplasmic reticulum, found around the perimeter of the myofibrils. So I'll make the myofibrils now in orange here. I won't do all of them, but do a lot of them. So we're looking at the ends of these myofibrils, and we'll magnify them even more here in a minute to see what's going on inside of a myofibril. We're going to go ahead and label some of the things that we can label now on this diagram. First of all, the outer membrane here is called the sarcolemma. Plasma membrane on most cells is called a plasmalemma. So this is a sarco having to do with muscle. It's always sarco as a term that refers to muscle. There's a couple of nuclei in here. So we've got the nucleus. It's usually at the outer perimeter of the muscle fiber. I put in here a whole bunch of these little purple dots representing mitochondria. I put in some transverse tubules. Those red lines coming in here. The green lines represent the sarcoplasmic reticulum. Sarcoplasmic reticulum, and then the orangey dots in here represent myofibrils. They make up the bulk of the material that's in here. The way I've drawn it, I have a little bit too much space between them. That space where the sarcoplasmic reticulum and the mitochondria reside is really a very, very narrowed space, but in my artwork I had to make room for them in there. So they would really be squished in between all these myofibrils that take up most of the volume inside a cross-section of a muscle fiber. Now, if we're to look down inside of a single myofibril in a muscle, in a skeletal muscle, you'd find this arrangement of actin and myosin cut in cross-section. So each of these represents a long fiber. Each red dot represents a bundle of myosin molecules that are linear molecules that would run this direction. And each of the blue dots represents the actin molecule that runs this direction as well. So we've cut across them, but they set up a fairly regular pattern, not exactly completely uniform, but pretty uniform pattern of spacing with the actin between the myosin molecules. So in the red we have myosin. In blue, we have the actin. So what I have on the board here would be a small portion of a cross-section of a much larger field of actin and myosin within a sarcomere, in a cross-section of a sarcomere. So there's a large myofibril structure here, and around the outside of the myofibril There's not a membrane or anything like that, but there are transverse tubules. So this would represent a transverse tubule or T-tubule in the perimeter there. There's sarcoplasmic reticulum. So let's put in a little sarcoplasmic reticulum next to the T-tubule. And then let's put in a mitochondrion over here. So we have mitochondrion, sarcoplasmic reticulum, transverse tubule, all found bordering a single myofibril within a muscle. So hundreds of myofibrils within a cross-section of a skeletal muscle fiber and thousands of myosin and actin filaments within a single myofibril, each of them surrounded by scattered T-tubules, sarcoplasmic reticulum, and mitochondria in there. Here's another view of the inside of a skeletal muscle fiber. On the outside, we still have this sarcolemma. On the outside, this is a large nucleus over here. The nucleus toward the outside, toward the sarcolemma. These are transverse tubules. One here. And spanning between about the position of the transverse tubules, we have the single sarcomere units. In the sarcomere units, we have the actin and the myosin in red, actin in blue, and they would be tied to the next sarcomere unit and the next and the next. So there'd be hundreds and hundreds of these sarcomere units in a long line gathered into bundles, those myofibril bundles. So a myofibril is just a whole lot of sarcomere units end to end gathered into large bundles within the skeletal muscle fiber. So far here I can label the actin, the myosin, and on the myosin molecules I have these little flag-like little bumps on the ends here. We're going to call these myosin heads. So there's a myosin head, single unit there. There are several at the end of each myosin. At the end of your actin, you have what's called a Z line. How can I label this? This is our Z line here. And down the very middle between the myosin molecules, right along here, we have what's sometimes called an M line. M stands for a protein known as myomesin. In addition to these, we have something called a titin filament that attaches our, I usually draw this as kind of a coiled looking structure, zigzaggedy line, titin protein filament that joins the myosin to the Z line. So this is titin, T-I-T-I-N, titin. So those are the components of a single sarcomere unit. Single sarcomere unit. Now, the way these things work, once an action potential reaches the membrane, a depolarization wave hits the membrane it's going to travel down along these transverse tubules, and as it does so, it's going to encounter these spaces called, well, this is the sarcoplasmic reticulum we talked about earlier. So sarcoplasmic reticulum lies next to the transverse tubule, and that stimulates, once the action potential comes through here, that somehow stimulates the sarcoplasmic reticulum to release calcium ions. Calcium ions then surround all of the sarcomere units, and it causes a change in shape in the actin, the blue lines here, so that binding sites for the myosin heads become exposed. Once the myosin head binding site becomes exposed on the actin, the myosin head will grab and pull, and that pulls everything together to make a muscle contraction. So that's how muscle contraction works. Also in here you would find some mitochondria, and I didn't draw those in this picture, but they would be kind of distributed around the sarcomere units as well, so we can kind of draw one in here. Mitochondria. So large need for mitochondria in muscle and skeletal muscle to supply the energy demands of the contractile process. Okay, we've been looking at a muscle. We made a cross-section of a muscle belly earlier and looked at the components inside there, and then the microscopic structure of skeletal muscle fibers. Now we're going to look at the more gross structure, the larger structure of a muscle. Typically, muscles have a central region we refer to as the belly that contains all the skeletal muscle fibers, and then toward the end, they make contact with dense regular connective tissue in the form typically of a tendon. We'll talk about other names given to their attachments to a bone or other structures here in a minute. But anyway, so this is the basic structure, and so this is the belly of the muscle. One end is going to be more fixed in position and less movable, usually, and that is referred to as the origin. Less movable. And the other end is going to be more movable. And that's referred to as the insertion. The insertion of the muscle is the more movable end. In most every muscle, it's fairly easy to identify the more fixed end, the less movable end, as opposed to the insertion, the more movable end down there. So origin and insertion, two important terms to know when you're talking about skeletal muscle. Now, kinds of attachments. What I've drawn here are narrow attachments, what we would call tendons. Tendon is a narrow strap-like attachment. There's quite a bit of variation in how narrow they might be, but still be called tendons. But on many of our muscles, we have a very broad attachment of one end of the muscle, what we would call an aponeurosis. A broad attachment of the muscle. So muscles along your back, your latissimus dorsi, for example, has a long sheet of connective tissue that attaches to it from which it pulls. So anyway, aponeurosis. Sometimes there are muscles let me move my camera and explain this a little bit. Sometimes there are muscles that join end to end without joining to another bone. So you'd have a muscle belly, and then another muscle belly, and then another muscle belly, like this, with connective tissues in between. So connecting muscle to muscle in a series like this is what we call a tendinous inscription. And in your abdominal muscles, your rectus abdominis, right down the middle of your belly, to get those six-pack abs, you have a belly, then connective tissue, then another muscle belly, and so on. The tendinous inscription is that connective tissue between one muscle belly and the next in a series. So those are types of muscle attachments: tendons, aponeuroses, tendinous inscriptions. Okay, we're talking about muscle tissue. So why do we have an embryo on the board? That's the question. Remember when we were talking about the formation of the neural tube, how we formed neural crest tissue up above the neural tube after the neural tube closes in, so that just above the brain and spinal cord, all along here, is some neural crest tissue. Also, we had the formation of somites from the mesoderm just next to the notochord, all along through here. In the head region, we don't have the somites grow into muscle-forming tissues. So somewhere up in here, there's an otic capsule. Let me go ahead and put that in here, an otic capsule, where the ear will develop, the inner ear structures will develop. Mostly in front of the otic capsule that develops in here, we have we have neural crest tissue that we call head mesenchyme. That will contribute cells that will diversify and become most of our musculature and skeleton and connective tissue around our developing brain up here, between the surface ectoderm on the outside and the brain in there. So anyway, we have a lot of tissue coming around here from that. Then starting behind the otic capsule, we have more definition of our somites. Our somite blocks form along here, and they will contribute mesoderm for the rest of the musculature through the rest of the body. So up here in the front of the body, we have two kinds of musculature that we derive from head mesenchyme. First of all, there's that forms around the eye muscles. These are the extrinsic eye muscles. Remember, intrinsic eye muscles are from smooth muscle. So we have extrinsic eye muscles that move your eye in the orbit, so you can look left, right, up and down and all that, moves your eye around. So extrinsic eye muscles. The rest of this musculature in here becomes what we call branchiomeric. So all this muscle comes in here and becomes associated with your visceral arches. Mandibular arch, hyoid arch, branchial arch 1, okay? So anyway, we've got this muscle coming in here from in that area. It's all neural crest that became head mesenchyme, forming branchiomeric muscles. Our chewing muscles, our facial expression muscles, some of our throat musculature is derived from branchiomeric musculature. Now, behind the otic capsule up there, some of these tissues come around up under here and become what we call hypobranchial muscles. So these are from somites that developed in this area. So they're from true mesoderm, not from neural crest. And these contribute to muscles primarily of the tongue. But muscles in that general region, they come up from behind the branchial structures and move in to form the central tongue muscles and maybe a couple of other muscles. They're associated with the hyoid. The remainder of the musculature is going to come from the remainder of these, from the somites. Okay, so we're identifying places of origin for our different muscle groups in the body. We've already mentioned that head mesenchyme from neural crest forms extrinsic eye muscles and branchiomeric muscles. Those are associated with the pharyngeal apparatus and some of our facial muscles. Now the somites from the body, mesodermal somites, contribute to most of the trunk and what we call hypobranchial muscles that come up and form the tongue and some of the associated muscles there in the very center under the tongue. So anyway, we started with those, and then we said we needed to do something with the trunk, the remainder of the trunk musculature. So if this is a vertebra here, these are the transverse processes of a vertebra. They kind of set up an imaginary line, if you extend that out, that divides that back musculature, the trunk musculature, into two groups. One muscle group that's found above that line, up in here, this back muscle. Here, this is referred to as epaxial. The muscle that's formed below that line, that usually goes out around the body even further, out in here, this is called hypaxial. In a shark and in fish, this epaxial group would extend further out along the body. In mammals, the epaxial muscle is pretty much isolated very close to the vertebra without much of it extending out very far from that transverse process out there. Most of your trunk musculature in the human is going to be from hypaxial. Only that right along the middle of the back is epaxial muscle in there. So if we put in like a dotted line out here, this is real prominent in fish, this separating line between epaxial and hypaxial. Not so much in higher organisms, but anyway, where that appears, it's called a horizontal skeletogenous septum, or just a horizontal septum. Horizontal skeletogenous septum, or simply a horizontal septum. Now below this and inside here, this would be body cavity all in here. There's your body cavity. There's your muscle layers between the body cavity and the outer body wall. Our last muscle topic to contend with here, the appendicular muscles, going into your appendages. There's quite a number of them in different groups with different names, so we won't get into too much detail here. We're just going to mention that some of the muscles extend from your axial skeleton to your appendicular skeleton. And we call those extrinsic appendicular muscles. So you have your extrinsic appendicular group. So something like your pectoralis major that attaches to your sternum and ribs and reaches over to your arm at your shoulder. That would be an extrinsic appendicular muscle. Your latissimus dorsi that runs from the middle of your back up to your humerus. Those that attach from your back to your shoulder blade over there would be considered extrinsic. All the rest of them that run from your shoulder girdle to parts of your arm or completely within the arm, those are considered intrinsic appendicular muscles. All of these are derived from the trunk mesoderm, as was your epaxial and hypaxial muscle of the trunk. So anyway, this is all from somite. It's all mesodermal in origin, the appendicular muscles. They're either intrinsic, they're completely within shoulder girdle and limb, or pelvic girdle and limb, or they're extrinsic. They have their origin outside and then they extend to the girdle or to the limb in there. So a quick review of what we've just been over. First of all, we were looking at our major skeletal muscle groups, kind of outlining them. We talked about there being some that were derived from head mesenchyme and some that were derived from mesoderm from the somite mesoderm in the trunk of the body. So the extrinsic eye muscle and branchiomeric muscle were from head mesenchyme. Starting behind the otic capsule, we saw hypobranchial muscle that came into the tongue, epaxial and hypaxial of the trunk that are divided from above the horizontal septum and below the horizontal septum, epaxial and hypaxial. And then the muscle that goes into the limb is either considered extrinsic if it has its origin in the axial skeleton and extends to the appendicular skeleton, or if it's completely within the appendicular skeleton from girdle to limb or within the limb, it's considered intrinsic appendicular muscle. Those are your major skeletal muscle groups. Two other curious things worth noting before we leave the muscles. There are some mammals that have a significant amount of what we call cutaneous muscle. Cutaneous muscle. Muscle in the body, in the skin, not attached to the skeleton, that usually occurs as large sheets or broad sheets, very, very thin layer within the dermis of the skin. The best example of this is if you ever see a horsefly land on a horse or something irritate the skin of a horse. The horse will shake its entire skin. It has the ability to contract that muscle and just shake the skin. So that's something that's very often observed when you look at horses. They'll do that twitching motion on their back, on their skin, on their sides. So cutaneous muscle, we have very little cutaneous muscle. When we do our cat dissections, we usually see some cutaneous muscle if we are removing the skin from the cat. So anyway, very, very thin sheets of muscle found in the dermis of the skin. We have some just along the side of our face and neck here. So cutaneous muscle is an interesting and odd thing. Another odd thing with this skeletal muscle electric organs. This is best developed of course, in the electric eel, but some other fish also have electric organs that will generate a pulse of electric current. Basically what happens here, instead of being a muscle that's filled with contractile elements, they just have a whole lot of muscle, skeletal muscle organized into membranes that will transmit action potentials very rapidly across many membranes stacked together so that you get this large current. This large movement of ions in the fish generates a large electric current pulse in the water around them. So electric organs, where they occur in these animals like the electric eel, they are modified skeletal muscle. Beyond the electric eel, there's an electric catfish, a few other kinds of fish that generate short pulses of electric fields for defense, but then there are some that generate electric fields that they use to navigate, to detect materials around them in the water. Just very weak electric fields that they can generate. So anyway, electric organs, when they're present, doesn't do any good if you're not an aquatic organism. When they're present in fish, it's derived from skeletal muscle.